Hi, my name is Camila Guerrero, and I work in Bruno Paiva's lab here in Pamplona, Spain. I would like to thank the IMF for the kind invitation to share our results here in this webcast session. This year at ASH, we presented our work titled Multiomics Profiling of Measurable Residual Disease for Understanding the Biology of Ultra-Drug Resistance in Multiple Myeloma. The persistence of MRD is one of the greatest challenges in multiple myeloma. In fact, we still don't know why these few tumor cells are empowered to resist treatment, why additional therapy using the same drugs is not able to overcome persistent MRD, as was shown last year at ASH from results obtained in this Spanish trial, and why within patients with persistent MRD, time to disease progression varies, and even a few of these patients remain progression-free for longer periods. Given all these uncertainties that remain unanswered, we aimed to understand MRD resistance through the multiomics profiling of patient-paired tumor cells at diagnosis, MRD, and relapse after different treatment modalities. This study included 118 patients, 28 transplant ineligible elderly patients enrolled in the GEM-Claridex trial, who received lenalidomide and dexamethasone plus or minus clarithromycin, and 90 transplant-eligible patients, with a small cohort of high-risk smoldering myeloma patients enrolled in the CSAR trial, and a larger subset of patients with active multiple myeloma enrolled in the GEM 2012 trial. Both studies consisted of induction, followed by autologous stem cell transplant and consolidation prior maintenance. A total of 289 bone marrows were sequenced, and we would like to stress that all MRD samples were matched with tumor cells at diagnosis. Accordingly, there were 96 patient-paired samples post-induction, 34 post-transplant, 13 post-consolidation, and 28 that were collected at diagnosis with at least one MRD sample and at relapse. Patient-specific aberrant phenotypes were used to isolate tumor cells by FACS, and MRD was detected by next-generation flow cytometry. The co-isolation of RNA and DNA was performed prior to RNA and whole exome sequencing. Gene expression analysis revealed only 6 genes to be deregulated in MRD cells compared to tumor cells at diagnosis at all time points. But more interestingly, only a few genes were deregulated in MRD cells post-induction, while a much higher number of genes were deregulated in MRD cells post-intensification, especially post-high-dose therapy and transplant. These results suggest that transcriptional MRD adaptation might peak after this time point. Findings from RNA-seq resonated with those obtained from whole exome sequencing, and we found a median of approximately 40% mutations shared between tumor cells at diagnosis and MRD cells post-induction, while we only saw less than 15% mutations shared between tumor cells at diagnosis and MRD post-intensification, with a higher number of de novo mutations in MRD cells. As an interesting example, this patient had a de novo TP53 mutation, here in yellow, that was observed in MRD post-transplant. Interestingly, this patient never achieved undetectable MRD with maintenance therapy and actually relapsed soon after. When studying recurrent mutations, or those mutations that were found in 5 or more patients, we saw that KRAS was the most recurrently mutated gene. But interestingly, the gene BCOR-L1 was only recurrently mutated in MRD cells. When studying the expression of this gene at diagnosis in 2 cohorts, the GEM 2012 trial of active multiple myeloma patients and the COMPASS study, we saw that high expression of BCOR-L1 was associated with worse outcomes according to progression-free survival. We then investigated if the transcriptional mode of MRD resistance was associated with subsequent disease progression, and we found biological processes such as cell activation and regulation of immune system processes to be significantly enriched in differentially expressed genes from tumor cells at diagnosis versus MRD from patients who relapsed versus those who did not. Interestingly, as preliminary analysis from tumor cells at diagnosis, MRD, and relapse, we saw that the MRD clone clustered further away from tumor cells at diagnosis and relapse. Finally, whole exome sequencing results resonated once again with those obtained previously from RNA sequencing, and we saw a higher number of private mutations in MRD cells, here in blue, and in tumor cells at relapse. Indeed, both RNA sequencing and whole exome sequencing data suggest that MRD could represent a singular genomic state between continuous genomic evolution from diagnosis to MRD and relapse. As another example, this patient had a TP53 mutation at diagnosis that became undetectable post-induction, reappeared post-transplant, and finally became dominant at relapse. Finally, as key takeaways, we would like to stress why this abstract is important to the myeloma community. And as such, we presented the largest data set of longitudinal MRD biology profiling in multiple myeloma. We believe it is important to understand the biology of this clone to identify new drugs to overcome MRD resistance. Our results reveal transcriptional stability during induction with relative divergence after transplant, as well as progressive genomic evolution that peaked after this time point. These findings could help explain the continued responses with longer induction regimens and limited efficacy of consolidation after transplant that was observed in recent studies. And finally, the transcriptional and genomic singularity of MRD cells when compared to tumor cells at diagnosis and relapse suggests that using drugs with alternative modes of action are needed to overcome MRD resistance. I would like to thank all the patients and caregivers, the University of Navarra, Consortium Euroflow, and the PETEMA Foundation, and last but not least, my colleagues, for giving me the opportunity to present these results to you. Thank you very much.